Good morning, AP students. So today I believe it's the 7th lesson, and it's a mini lab that we ordinarily would do in the lab, and it is on cell size, and the concept behind the lab is actually pretty basic. So the rate of osmosis and diffusion is important to a cell. So if you have a newly cell formed from mitosis, then it's fairly easy for anything inside of that to diffuse because the cell is small. And as the cell enlarges, the problem comes in that the rate of diffusion doesn't change. And so that's one of those things that's difficult for a student to get over their head. So a thought of that is if I am walking down a hallway and I have a set pace, and so if I'm going down a short corridor, it would be how long it would take me to get that to the end of the corridor. So if you think of upstairs in the new wing, for me to get from one end to the other would take me a certain period of time. But if I'm doing a fire drill and I'm leaving my room and just going to the stairwell, that's a much shorter distance, so it'll take me less time to get there. So that's the problem with diffusion. It's a set rate. It doesn't change. So if the cell is very small, it takes a short time to diffuse. If the cell has gotten very large, it takes a long time for diffusion to happen. And that puts difficulty on a cell. And it's one of the reasons why cells divide, is that they simply get so large that the rate of diffusion is too long, and the cell actually gets a command to go ahead and divide, and it should do that. Or it should kill itself through apoptosis. And so here we're going to go through a very basic cell lab. And I have a video for you to watch, and I wanted to give just a brief explanation of that video. The teacher is actually very good. I make fun of him in that he's Mr. W, and in his photosynthesis discussion, he plays a song, and so he's known as also as a singing biology teacher. And in that song, he's doing it in a closet back in the early days of Screencastify, where they were worried about what the background is, etc. And so he's sweating profusely. And so it's just, he has a very good song to sing, but it's just kind of gross to watch. So I have that video of him doing the lab because he really does a good job in it. I just wanted to give a little bit better explanation of a couple components because he sort of flies through it. And so, first of all, he calls it agar, and I pronounce it agar. And so agar is what we would have used in CP Bio. It's dried seaweed, and it is sort of like Jello, in that it's a clear substance or a slightly tannish colored substance. And it's in a powdered form, and adding distilled water to that, you can heat it up and then put it into Petri dishes, and that's what we would have grown our bacteria on when we did the gram staining lab, when we took the samples of water from out in Beaver Brook last year. So in this case, they're adding a chemical, phenolphthalein, and you would have used that in chemistry if you had Mr. Moore. He used it as some sort of indicator. I don't know why. It could have been that he was just showing you the difference between an acid and a base. But I remember seeing the bottles of it in the lab, so I know that one of his classes at least used phenolphthalein. So what they've done is, and if we were doing this lab in our lab, we would be doing the exact same thing. We would be taking, in this case, I would take a square trough instead of a Petri dish, and I'd be pouring the agar in that, but I would have added phenolphthalein to our agar solution when it was still liquefied. And if I was a lab tech, probably, and especially working in a bacteria lab, I probably would mix together sheep's blood to some of those Petri dishes, because bacteria particularly likes to grow very quickly on sheep's blood. So this is just one of those things to understand, that we can add a lot of different things to agar depending on what use we're going to have. And so what you may not have been aware of is that when I was making those Petri dishes for your bacteria cultures, in fact, I think one class made their own Petri dishes, we added some beef broth to that because that's another thing bacteria likes to grow on. But in this case, the agar is distilled water is added to that powder, and then the chemical phenolphthalein, and then put in a square trough, like a very small bread tin, and then that's allowed to harden. And then that is cut into strips and squares. so that you have a variety of sizes. And then taking a ruler, that agar cube is cut down into a 0.5 centimeter cube, a 1 centimeter cube, a 2 and a 3 centimeter cube. And I should have put down centimeters for this. And then those cubes are dropped into a solution of vinegar. And vinegar, acetic acid, is, as the name says, an acid. So here you're going to see that over a period of time, the vinegar diffuses inside the agar cube. So here I'm going to have a 0.5, a 1 centimeter, a 2 centimeter, a 3 centimeter. Didn't think that out very well. And the cubes are allowed to be in the solution of vinegar for 6 minutes. So part of the lab we would have done is that we would have left it in longer. So at 6 minutes, we would have taken out the 0.5 centimeters. At 7 minutes, we'd be taking out the 1 centimeter, etc. And we'd ask 1 or 2 lab groups to sort of play with their own time. So we would have one lab group that was taking it out every 2 minutes, and would have another lab group choosing to do it every 4 minutes. So that what we could get was a nice graph based off of time and your cubes and whether or not they had any phenolphthalein in it, left in it. And you will see that at the end of the 6 minutes, the 0.5 centimeter cube has no phenolphthalein left in it, which means that vinegar has all the way diffused through that agar cube. And remember, this is not a cell, this is just a chemical. And then the 1 centimeter, would have been interesting to see how long it took before that diffused all the way through. And then again, 2 and 3 centimeter cube. So what I want you to do is, now that you understand the basis of the lab cell size, now we have to understand why is AP College Board wanting you to do this lab, or at least look at the lab. And so the reason behind that is that to understand that diffusion is at a set rate. It doesn't change in a cell. So a small cell, it's going to diffuse quicker, not because it's speeded up, but because it has less distance to go. And in a very large cell, the diffusion rate is going to be very long. And so it has to do with surface area as well as the volume that's in that cell. So some cells have tricks to increase the surface area of them. For example, if we take a look at a neuron, here we have little, they're called dendrites, or little hairs off of that. And we have the neuron body here. And then we have a long, like a stick that's called the axon. And then we have these little terminal buds here. And so what has happened is with these branches is that the surface area has increased greatly. And when the surface area increased greatly, it allows for the diffusion to happen a little bit better. And so there are an awful lot of tricks a cell has to increase that surface area. For example, if I draw another cell here, or the flagella, the cilia, or the little branches off of this, all increase surface area. And one of the things that almost everyone in bio brings up is elephants and how elephants changed through evolution. And when we look at what are called cladograms, or evolutionary trees, where organisms have an ancestor, a common ancestor, and then there are branches off of that. When you look at the elephants, there is a change in their ear structure. And that over, hundreds of years, thousands of years, their ears, they're actually get much, much, much larger. And so that is due to habitat, where the elephant once had quite a broad place that it lived, and of course ancestors, the mastodons and the woolly mammoths. I have a deciduous, or a baby tooth of a woolly mammoth that I found in my backyard in South Carolina. So after the time period of when the ice age ended and the ocean waters were receding, where I lived at one point had been ocean, and then later on was land, and someday a baby woolly mammoth was walking through my backyard and lost a baby tooth, and I found it hundreds of, well, 1,000 years later or whatever. So that's one of those things that if you look at the fossil evidence, that ear size has increased. So if you think of the elephants having been trapped when some of the land bridges finally broke out through Pangaea, then they were trapped in an area where it ended up becoming very hot and arid. And so the only way those elephants survive from an evolutionary standpoint is for the surface area of their ears to get larger and larger to help diffuse the heat off of them. So they were increasing the surface area to help with diffusion. And so take a look at this lab, and then I have a short assignment with the lab for you to do as well, a short discussion piece. Thank you.